Okay, hi everyone. So today we are going to wrap up our discussion about B cells and how their production of immunoglobulins is so important for our immune system. We will revisit this topic later as we begin to put many of the things that we've learned up until this point in the semester together in functional examples when we look at different types of conditions, such as allergies, autoimmune diseases, immunodeficiency diseases. Not surprisingly, we'll be talking a lot about things like antibodies, B cells, T cell receptor. But for now, this lecture is going to kind of wrap up our discussion about certain types of mechanisms that specifically get us appreciating how B cells and antibodies play an important role of our immune system. So you will have a quiz on this stuff due this week, Thursday night, as usual. Okay. So let us sort of recap what we've been talking about in terms of the importance of antibodies. Remember, antibodies are the soluble or free-floating version of immunoglobulins that a B cell can make if that B cell has been selected because its immunoglobulin is of a particular recipe that makes an IG shape that binds something like pathogen successfully. And then that B cell may be encouraged by things like helper T cells to multiply so that your body now has many, many copies of this knowledge, right? Many copies of identical B cells that each possess the same recipe for the immunoglobulin. Some of those will be sent up into the germinal centers of our lymph nodes to undergo hypermutation to try to make the IG recipe better and better and better so that you get stronger and stronger binding immunoglobulins. Other clones of that B cell may be encouraged to become plasma cells so that they then will switch from the production of the membrane-bound form of the IgM that they started with to now maybe the free-floating form of the IgM antibody, right? And we said then that over time, as the B cells in your lymph nodes are undergoing this affinity maturation, the hypermutation that leads to better and better quality immunoglobulins, that you would then get wave after wave of plasma cells leaving your lymph nodes, sending out wave after wave clouds of better quality antibodies. Right? And these antibodies, since they are free-floating little proteins, right, have many advantages as a powerful immune effector compared to when you're just talking about cells themselves acting as effectors, right, like macrophages or natural killer cells, etc. We said that the advantage of having antibodies is that they can swarm. They can swarm and cover antigen targets. They can neutralize pathogens by blocking the ability of pathogens to interact with host cells in your body, for example. So viruses cannot gain entry and infect more cells of your body. They can deliver pathogens to other effector cells of our immune system, which we'll take a look at today. One important way that antibodies work is they amplify or enhance the function of your complement proteins. Remember, at the start of any infection, your innate immune system has sort of first crack at it, right? Because your complement proteins, macrophages, most of the time are pretty successful in opsonizing bacteria or viruses and then phagocytosing or destroying them. Right? But in certain circumstances, if the pathogen is different enough than something that your innate immune system is only pre-programmed to recognize certain types of features. If you have a new virus, for example, a new pathogen, that is bad clown, right? And so if your innate system does not succeed in defeating the infection within a few days, then by then, the pathogen, like bad clown, really begins to make you sick, right? Because the pathogen begins to multiply and all of the terrible side effects of your body's resources being used up and compromised are really leading you down the path to dying, right? Unless your immune system can come up with a way to deal with this. And so that's why B cells get selected, right? And then eventually you'll have the release of these antibodies, right? And one of the things that antibodies do is because they were chosen to be able to bind to the novel pathogen, right? Whereas previously you didn't have anything that could bind to it very well, but now these antibodies do. They can also help by recruiting the complement proteins and getting the complement proteins to stick much better, right? Thereby sort of like reintroducing the role of your innate immune system in destroying the targets. And importantly, we said your adaptive immune system has this feature of being able to have immune memory because your body came up with selecting these B cells and encouraging these B cells to refine themselves to make the best quality immunoglobulins that they could, right? Whatever you're left with at the end of that infection, your body can remember that, right? In the form of some of those B cells and some of those plasma cells sticking around, right? So that in the future, if that same pathogen comes back again, you don't have to start from square one. You start off with already having really good immunoglobulins available. Right? So those are important things to keep in mind. And that's why, the evolutionary invention of B cells and immunoglobulins is such a big deal, right? And I wanted to remind you that not all life on Earth, right, can do this. Right? It's really only in the tree of evolution from vertebrates onwards, we have the ability to do this. Right? Okay, so when we talk about the involvement of antibodies, right, as being a important part of your immune defense that ends up saving your life, right, because it was able to actually defend against a novel threat, there are two ways or two scenarios where you can end up with these antibodies. Actively acquired immunity means your immune system actively had to be involved in making these antibodies. So that means everything from selecting the naive B cell clones that just so happen to have close enough fit IGs to start with, through the process of encouraging those B cells to hypermutate, right? And then having those memory B cells stick around with the best recipe that you came up with following your illness. That's a very active process. Right? So your own immune system responds to antigen that's presented to it by producing your own version of antibodies. So in the natural world, this would happen from being exposed to bad clown, right? I mean, I hate to break it to you, but that's kind of how we evolved, right? Bad things oftentimes have to happen in order for our immune system to actually develop immunity. So you get infected, but you're able to beat the infection by making these antibodies. So that's actively acquired. Right? The other way that is afforded us by modern medicine is through vaccination. Right? And we will study vaccination and vaccines in more depth after spring break. Okay. But for now, let us just say that vaccines, the purpose of vaccines is to stimulate your immune system to actively participate in making antibodies as though it was a pathogen that is really infecting, right? So the process of responding to a vaccine and getting immunity is still considered actively acquired immunity. I think that throws a lot of students off because you think, yeah, I get how active acquired immunity naturally is, like I get sick and then I learn how to make antibodies, right? That's easy to imagine. That's active. But you think, well, if I get a vaccine, like a shot in my arm, I didn't really do much. I just sat there and I got a shot. And now, oh, now I'm immune, right? No, you forget, right? It's still up to your immune system to respond to that vaccine and make your own antibody. So it still is actively acquired. Okay, so keep that in mind. That's important. Right? On the other hand, passively acquired immunity is when you actually get antibodies already pre-made from an external source. So in modern medicine, we have things like antivenoms. So a lot of times if you get bit by a snake or something like that and you're poisoned, or bit by, some nasty kind of poisonous insect, right? And your body, is having a response to some sort of toxin, And the venom, oftentimes anti-venom that's given to you at the clinic, right, is actually an infusion of antibodies that have been designed already, right, and manufactured already to target the venom. Right? And we will look at that in today's slide. Right? Other types of things that are becoming more popular are immune therapies. So many very high profile types of diseases now, especially things like cancer, right? That, we will talk about cancer at the end of the semester, but for now, hopefully it's not too far-fetched for you to imagine that the fact that if you develop cancer, that actually represents somewhere along the line your immune system failing to protect yourself, right, against the cancer cells. So immune therapies that involve infusion of high quality purified antibodies that can target things like cancer cells are becoming more and more popular. Right, but those are kind of artificial. Those are man-made modern medicine. Are there any examples of passively acquired immunity in nature? Right? Absolutely. Right? Because one of the most fundamental components of our immune system that we start our lives with is the passively acquired immunity for against various types of pathogenic threats that we get from our mom, right? As we are still developing in the womb and immediately after birth, as we are breastfeeding, right? So those are situations where you're actually getting antibodies directly from mom, right? And those antibodies go into your system and they are there to protect you. So we will end today's lecture revisiting that and taking a closer look at that. Okay, so important. Actively acquired immunity versus passive, right? You have to be able to keep those straight. So let's look at some examples here of actively acquired immunity first. Right? So here is a natural example. Here we have two brothers, a big brother and a little brother, and they are both exposed to strep throat, right? Let's say that their cousin comes over to visit and play, right? And their cousin is coughing, has a sore throat, cousin has strep throat, and it's cousin, because it's like three boys, they just like breathe on each other, right? Anyhow, so the strep throat, I think we've all had strep throat, right, is caused by different types of pathogens, but one of the one that's most common classically is the Streptococcus bacteria. So in this case, big brother has had strep throat before, right? He's previously, his immune system has previously encountered this Streptococcus bacteria before. So when he breathes it in, he already has the memory of this in the form of what? He has B cells that have stuck around since the last time he had strep throat. And those B cells have the knowledge how to make the highest quality immunoglobulins that his body, his immune system came up with, right? When he successfully beat strep throat the previous time, whether it's a few months ago or potentially even a few years ago, right? So those memory B cells, the moment he breathes in the bacteria, because his cousin coughs on him or spits on him, whatever, right? And these bacteria are going down his esophagus or trachea. I think this is his pharynx, so maybe it's going down his trachea, right? His memory B cells that he has present begin to detect, again, that here is the return of that pathogen that they have memory about how to make their immunoglobulins to, right? So bits and pieces of the bacteria are presented by professional antigen presenting cells, right, in brother's body. And so therefore in places like the lymph nodes, places like that, where he has some of these memory B cells, they get stimulated again, right? And they wake up and right away, they start things going again, right? They very quickly start releasing antibodies. And these antibodies would be in the form of IgA, because IgA is the form of antibody or immunoglobulin that is most widely used in the mucosal layers. So the layers of our respiratory tract that have a thin layer of mucus on them, right, these IgAs, and they function as dimers. Remember, they look like little bow ties because they are held together on their stems. So Big Brother, right, is able to have production of high quality IgAs based upon his B cell memory of this from before. And so they sort of neutralize, they cover up the bacteria binding to the antigens. And as a result, the bacteria do a poor job of clinging to his epithelial cells and infecting his epithelial cells, right? So the majority of them, you can see here, just end up going down, like going down his esophagus or whatever, and they're eventually destroyed in his gut, right? So they're not able to actually infect or start a big infection again. However, little brother here, right, he's never had strep throat. So he's never experienced these antigens before. He has no antibodies or no B cell, memory B cells that know how to make these antibodies. He has to start from square one, right? But unfortunately, that involves what? His innate immune system not being able to defend against these bacteria very well. So these antigens on the bacteria, maybe these are little proteins that are involved in sticking to surface features of his epithelium, respiratory epithelium. They gain entry into his cells, right? And they infect and they begin to multiply. And this means that for the first, 2-3 days, maybe little brother doesn't experience anything funny, but over time, right, he begins to experience the symptoms: sore throat, fever, etc., inflammation, right, as the bacteria are causing his epithelial cells to be damaged or destroyed, right? So it might take a week or so before his adaptive immune system, right, selects brand new naive B cell clones, right, and goes through that whole process. But hopefully, hopefully he'll come up with something, right, and then he will get better, right? Because I think all of us who had strep throat before, I don't think we ever thought while we were having strep throat that we're really going to die. I mean, think about it. When we were little kids, you don't really even know what's happening to you, right? But when you think about it in hindsight, right, it's not like you're really worried that you're going to die of strep throat, right? But that's only because your adaptive immune system finally woke up and made the antibodies to be able to save you. Because otherwise, I hate to break it to you, you would have died of strep throat, right? Importantly, what? Now, little brother, because he beat the strep throat, he will be like big brother, right? Next time they are exposed, like a different cousin comes over and sneezes and spits on them with strep throat, right? They both now have memory. But remember, the interesting thing is, right, the particular antibodies that big brother makes to defend himself with would be potentially different, right, than the antibodies that little brother made, because it all had to do with whatever random B cell clones they happen to have in their body as the start of the B cell selection process. Okay, interesting to think of. So this is the way Mother Nature built us to be able to use our actively acquired immunity to protect ourselves against things in the future. What about vaccines, though? So here's another scenario, right? So both of these scenarios, the brothers and also these twin sisters, these are scenarios that your textbook use. I think they're kind of funny in a way, because I don't know why, in a way, the book goes to the extent of actually coming up with like a story like this, but it's kind of nice. So you have these twin sisters, right? So they're in college together, and the flu shot becomes available on their campus health clinic, right? And so one of the sisters, she's really good. She says, I'm going to go get my flu shot, right? So she gets her flu shot and she's like really happy about it here, right? Her twin sister, unfortunately, she doesn't have time. Maybe her classes are really bogging her down. And she says, well, I don't have time this week, maybe next week, right? I'll go, And then guess what happens, right? The twin sisters, they hang out together, they live together, and they're with the same friends. So then the flu virus comes along, right? And so see over here in vaccinated sister, right? In response to the vaccine, she actively acquired immunity. Her body selected for B cells that made IG, right? And those IG are now part of her memory B cells, so that when she breathes in the real influenza virus, the flu virus here, right, her body starts producing high quality IgAs that bind to and neutralize the virus. And as a result, the virus is not able to gain entry into the cells of her respiratory system, right? And vaccinated sister remains healthy. However, on the other hand, right, that same cloud of virus, whatever, gets also breathed in by twin sister who didn't get vaccinated, right? So she has no immune memory of this particular antigen. So they've labeled this antigen here. These are the spikes on the virus. They're just referring to it as a hemagglutinin. Hemagglutinin is kind of a generic sort of label for any type of an antigen that can be used diagnostically to cause things like cells to coagulate, right, or agglutinate. But anyhow, it turns out that these spikes are the spikes that the virus uses to bind to certain surface features of our body cells, like these little sugars, these oligosaccharides that are on the surface of the respiratory cells. And unvaccinated sister, right, she becomes infected. So the virus gains entry, the virus releases its genomic material into the cell, hijacks her cells, and now her cells start making tons and tons and tons of more virus particles. And twin sister gets severely sick for 2 weeks as a result. I think this is kind of funny because they make it very dramatic, right? It's not just she gets sick, she gets severely sick for 2 weeks, right? It's like they're trying to teach you a lesson here. Why does the story circle around twin sisters, right? I think because they want to show you, the point is, these two individuals are genetically identical, right? They should have the same immune system and therefore the same robustness of their immune system to fight against this virus. The only difference here is one of the sisters, right, got a vaccine that allowed her system to already, or her immune system to already have a chance, like a sneak preview of the virus, so that she was able to develop immunoglobulins in anticipation of today when the real virus came along, right? But twin sister did not. So this case here involving a vaccine and the previous case of the brothers involving a sneezing, spitty cousin who gave them strep throat, right? They're both examples of actively acquired immunity. When we talk about immunoglobulins having the ability to neutralize certain things, it's not just pathogens that they can neutralize, right? Because an immunoglobulin is only binding to some three-dimensional shape that fits in its antigen binding arms, right? It doesn't mean that that thing that it binds to, that the immunoglobulin somehow knows that it's, oh, this is a virus, or, oh, this is a bacteria, right? It doesn't know anything. Igs are just little protein molecules. They just bind whatever fits. So that means the thing that they may bind to might not actually be a pathogen at all, but it could be some sort of toxin, right? Some kind of a poison. Consider this. Many of the toxins that bacteria secrete, right, are actually small little proteins, small polypeptides. Right, so we are talking about things like, for example, salmonella poison, right, from food poisoning. What is it that actually makes you sick? A lot of the things are these toxins that are released by the bacteria, right? And you think like, well, why would the bacteria want to do that? Well, it's for that whole reason. It's poisoning your body, right? Because it is actually trying to modify the tissues of your body, for example, to fend off things like your immune cells and to also gain access to lots of nutrients in your tissues. So many of these toxins are small proteins. Some of them are made up of different parts to it. For example, this toxin here, let's pretend it's made up of 2 parts, right? Both of these are protein parts, but they work together. One part, this sort of yellow structure here, is the part that's responsible for being the right shape to bind to, let's say, the surface of your cell, right? The other part, the red dot, is the actual toxic peptide. This is the part that if it gets into your cell, it can wreak havoc. So let's look down here in the cartoon. So this toxin, right, which is a protein polypeptide, or maybe 2 polypeptide chains together, right? The yellow part is the part that is the right shape to bind to this green protein that's on the surface of your cell membrane. As a result, it gets taken in to your cell, where it then falls apart. And then the red part then floats around inside your cytoplasm, wreaking havoc, right? Maybe it binds to things like, your ribosomes, maybe it binds to other components of your cells, and it poisons the cell. It prevents the cell from being able to function properly. So many high affinity immunoglobulin G and immunoglobulin A's work by functioning as neutralizing antibodies. What do we mean by that? Look over here. if your body has in it this blue antibody, right, that is the right shape to bind to the cell binding part of this toxic peptide, right? By binding, it would prevent this toxin from gaining entrance into your cell. In other words, it has neutralized the threat. So many types of antivenoms, for example, rattlesnake bites, right? I mean, we don't have rattlesnakes here in Hawaii. We actually don't have snakes other than the ones at the zoo or in people's homes or that have been let loose by people as pets, right? But, I think in movies, you've often seen that if you get bit by a rattlesnake, right, you have to get some sort of antivenom. Right? That's because the poison inside of the rattlesnake's venom is a peptide-based poison that gains entry into things like nerve cells, right, and prevents or poisons nerve cells, preventing the nerve cells from functioning, right? That's how the snake's venom immobilizes their prey, right? If the snake normally bites something like like another small mammal or something that it wants to eat, right? So if you get infused with high quality anti-rattlesnake anti-venom, that's usually, right, purified antibodies like this. That then once that's flowing through your blood, you usually get it infused through an IV drip and it goes through your blood, then it can bind up and neutralize the snake venom and facilitate its elimination from your body. Right? Many antitoxin vaccines, for example, the tetanus shot. I think most, if not all of you, have been vaccinated against tetanus. And then, every few years or so, you go to your doctor just for a regular checkup. Although, most of you, since you're college students, I don't know when the last time is you've been to your doctor, right? I know I never went when I was a college student. But anyhow, and then you're surprised sometimes. They're like, oh, you're due for your tetanus booster, right? And you're like, what? Right? So the tetanus shot is actually based around the peptide. It is essentially the yellow part of the toxin, right, without the dangerous red part. So when you go ahead and expose your immune system, right, to the yellow part, it gives your immune system a chance to actively start selecting for and having B cells, memory B cells that know how to make antibodies against this yellow part. Right? So that eventually, if you are infected by tetanus, then your antibodies can go ahead and neutralize the actual toxic version of it. Okay. We had discussed how one function of antibodies is to help out with the complement system. Right? So where you're experiencing an infection, where your innate system failed miserably to be able to opsonize and eliminate the pathogen. I mean, hey, that's the whole reason why you even had to wake up your adaptive immune system, right? So part of the way your antibody production works is it's going to now help out the innate system actually do the complement fixation that it failed to do before. So our first wave of antibodies produced when our B cells are selected during an infection, remember, are IgMs, right? This is what I call the quick and dirty first sort of wave of antibodies that's released, because this is all you have at that moment, right? Back at the lymph nodes, some of those B cells are already beginning to do hypermutation, but at least for now, this is better than nothing. Right? So some of that first wave of B cells start turning into plasma cells. They make IgM. And remember, IgM floats around as a pentamer, right? This shape here, where you have 5 IgMs held together at their stem. And they float through your blood in this planar form. And so each one of these antigen binding domains alone is pretty weak, right? It's not a good fit to the antigen, but it was good enough to actually form the basis of that B cell being selected, right? That knows how to make it. But since you have 10 of them in this pentameric ring, you can see here. So this big yellow thing is the surface of a bacteria, right? It kind of reminds you that a whole bacteria compared to something like a little ring of immunoglobulin proteins, right, is like massive, right? And on its surface are the antigens that this IgM recognizes. So the IgM binds to the antigens, right, forms this kind of platform or stable form of the IgM, and that recruits complement protein C1. The complement protein C1 binds to the IgM, and the tail of complement protein C1, this part up here that sticks up, right, recruits complement protein C4 and C2, and so that makes our classical C3 convertase, right? Because together the C4b and C2a, right, it attracts now complement protein C3 that's floating by. Previous to this, for some reason, whatever reason, the complement C3 was having a hard time getting activated and opsonizing the surface. But now, right, with this classical C3 convertase, you now start successfully being able to plant more and more of the C3b onto the surface. And remember, the C3b then will make it easier for things like macrophages that come along with the CR1 receptors on the macrophages to now bind this bacteria and phagocytose it, right? So this was the Velcro, right, with the ping pong ball and the beach ball. And we also learned, of course, that the C3b can also recruit C5 through C9 to make those membrane attack complexes and start making holes in the surface of the pathogen, right? This down here is kind of a neat cartoon. It's a little bit extraneous, but I think it's interesting. What it's showing is if you were looking straight down from the top, looking down at this IgM that's attached to the surface of the bacteria, now this gray thing is the surface of the bacteria, and you're looking straight down at it, you would see that C1, okay, so the C1 is the actual yellow part here, so they're not even showing the IgM because it's like tucked underneath it, right? The C1 gets recruited, and then the C1 starts recruiting this pink C4. So you then start imagining, you'd start seeing C4 molecules being recruited around here. And then once the C4 is constructed into the classical C3 convertase, it's going to start converting C3s and planting C3bs, right? So that's what these green things are that are beginning to then radiate out. Because remember, once you start making C3bs on its surface, then this forms the basis of making new C3 convertases, right? Like the alternative C3 convertase, where you'd have C3b recruiting factor B, and now you're making those types of convertases. So then the process starts radiating out, and the surface of the bacteria would quickly be covered in complement protein, right? And so the whole point is, like a week ago or something, when you were first infected by this bacteria, you were getting very poor opsonization of the surface by just relying upon C3 activation on its own. Now, a week later, after you've gotten really sick, right, now you start making IgMs that are helping this process happen, right? So now all of a sudden the bacteria are getting covered with the C3s, C3bs, and now your innate effector cells like macrophages can have a much easier time eating the bacteria. All right. Not only IgMs, but other Igs like IgG can also help with complement fixation. So for example, here's the surface of the bacteria. Here you have higher quality IgGs compared to the sort of lame, relatively poor quality IgMs that we looked at in the previous slide, right? Because now some time has elapsed. So back at the lymph node, B cells have been hypermutating, right? So now they are turning into plasma cells that make better quality Ig, and your immune system is not going to bother making IgM anymore, right? So those B cells class switch to make IgG versions of these immunoglobulins. And now you have tons and tons of these IgGs out there, and they also bind to the antigen, right? But maybe they bind much stronger now than the IgM did because their antigen binding domains have been refined, right? They've been hypermutated. So you can see they also recruit complement protein C1, so they can also help with the complement fixation that way. Importantly, the soluble IgGs in your blood, because they are individual little Y-shaped molecules, they have a lot more flexibility to them rather than the big pentameric IgM ring, right? So they can even bind up small pieces of antigen, right? So here's this, I don't know what this is here. If this is oligosaccharide, lipopolysaccharide, or maybe it's just a big protein, right? But the whole point is the IgMs can bind to this fragment, and they can do things like recruit the complement C1 to that. So that this piece of a pathogen begins to get opsonized to make it easier for things like macrophages to eat that, right? And you say, well, why would a macrophage, why is that important, right? If this is just a piece of a pathogen, right? Or let's say a piece of a toxin, right? Well, it's because, remember, if the macrophage is able to eat it through phagocytosis, then the macrophage is able to present it, right? Present it on things like your MHC proteins, so that then you can get your T cells kind of involved in the immune response as well, right? And any time you can get your T cells involved working alongside your B cells, you have a much stronger immune response. Okay. You can see here also, we have an example where the IgGs, because they are very flexible in terms of their hinge region, right? They can bind multiple instances of a small antigen, recruit complement protein C1 that way, and that would then eventually cause complement protein C3b to start getting stuck on the antigen. You know, it's kind of funny, right? Because at this point, since these are all just, you know, the antibodies themselves are just proteins, right? They have no way to kind of protect themselves against themselves being labeled with C3b, but that's okay, right? Because antibodies are expendable. You're making tons and tons of them. So you can see the C3b ends up being stuck all over this here. Then, you know, you have so many red blood cells, erythrocytes flowing through your blood, right? They have CR1 receptor on them as well. Okay, the complement receptor 1 that can then bind to the C3b. And now you have your red blood cells actually playing a role in your immune system. I mean, they're not doing much actively, because a red blood cell is nothing more than basically a sack of hemoglobin and some other proteins, right? But they're functioning as little transports now, right? Because they're carrying now all of this complement protein bound antigen to places like the liver or the spleen, where then the red blood cell, you know, might let go of it. And you have places, things like macrophages that can then happily take it from it. And again, this way the macrophage has a chance to process this antigenic signal and present it to your immune system, right? So it's a way of neutralizing things like, you know, potential toxins, and it's also a way of alerting your immune system to the presence of this. Okay? And it's all facilitated by the fact that you have these free-floating antibodies. So macrophages on their surface bind to the tails of antibodies using FC receptors. Remember, FC is that weird term that refers to the stem of an immunoglobulin, right? Because in the old days, when they were first trying to understand the structure, they saw that, you know, a Y-shaped immunoglobulin would fall apart into two small pieces and one big piece, the stem, right? So they called the stem part the FC, because that's the part that would be pretty much the same, right, from one immunoglobulin molecule to the next. You can't say the same thing about the arms, right? Because arms are all variable from one immunoglobulin to the next. But the stems, because they're all the same, they would crystallize, right? You could actually make a crystal of these. So they called it the FC region of the immunoglobulin. So receptors on the surfaces of immune cells that bind to or grab onto these FC regions, you know, not surprisingly, are called FC receptors, right? And depending upon which Ig isotype or class, whether it's M or A or E or G, right, we would just say that that particular receptor that the cell has, right, has an FC, for example, gamma receptor. So it can bind to the FC region of IgG, right? So yeah, this is what this is showing here. So this is the FC receptor, this blue part. You know, this isn't the entire receptor. You can see that this is part of the protein that just kind of ends here. Another part ends here. In reality, the protein would continue on in a big squiggly mess off to the left. But what they're just highlighting is this region here that binds to the FC or stem region of this immunoglobulin, right? So you can see the Y shape of this immunoglobulin. And I like this because it reminds you that it's not a rigid Y, right? It is this flexible, sort of flopping around kind of Y, right? Because it's a protein, right? Right. So here we have a macrophage with FC receptors on its surface. We have a bacterium, right? And as bacteria, your body has these high quality blue immunoglobulins, these IgGs, they're able to bind the surface of the bacteria, right, because they recognize the specific antigen that they were designed and selected for. And then because the macrophage has these Fc gamma receptors on its surface, it can bind to and hold on to the stems of the antibody, thereby facilitating, making it way easier for the macrophage to phagocytose and destroy this bacteria, right? So I want you to think about this. This macrophage could bind to this bacteria, right, without the involvement of antibodies. In fact, most of the time, that's how our innate system works, right? Our macrophage is pretty good at binding to the surfaces of bacteria, right? And if the surfaces of the bacteria are a little tricky to bind to, well, that's why we have complement proteins, right? So normally our innate system is the complement proteins coat the bacteria, and then you have the beach ball with the little fuzzy CR1, pads on them that can then bind to the Velcro, right? That's now stuck to the bacteria. But in certain scenarios, right, that alone might not be enough. And so in this scenario that's pictured here, what we're trying to appreciate is that it's only because your body knows how to make this high quality blue immunoglobulin. And this is what's making all the difference. And this macrophage is then suddenly way better at grabbing this bacteria and ingesting it. So when I say that, I want you to suddenly realize what I mean when I say that even though the antibodies themselves are very much a product of your adaptive system waking up, right? The fact that you have these antibodies now means the next time this red bacteria comes into your body, right, your antibodies will work with your innate effectors like this, the complement proteins and the macrophages, and greatly enhance the effectiveness of your innate system to battle it, even though the macrophages themselves are no stronger than they were before, right? The complement proteins themselves were no different than they were before, right? So it's not that the components of your innate system somehow work better. It's just they have the extra help of antibodies that together really boost their ability to fend off this bacteria, right? So that's what I said here. I should have put the text on the screen first, right? But that's what I'm saying. When you go back and look at the slide, think about this scenario, right? Because this sort of helps put together what we've been talking about and how it is that these antibodies in some way kind of can feed back and really help our innate system do a better job. And that's why it's important that we have memory B cells sticking around after we've beaten the infection so that they know how to release these high quality, in this case, the cartoon, these blue immunoglobulins, if they're needed again, right? Okay. And in italics, I said the next time the pathogen infects, like this cartoon above, those antibodies enhance the ability of complement and macrophages to wipe it out quickly because you don't have to start from square one again. Here are some other examples. So for example, natural killer cells, they have Fc receptors on their surface as well. So here you have a cancer cell, right? So this is a cell that's gone really bad in your body and it's diseased. And some of the hallmarks of cancer cells is they have really messed up protein production in them, right? Because all their genes are being all messed up. And so certain types of lymphomas, like B-cell lymphomas, you'll have a lot of CD20 protein on the surfaces of these cancerous lymphoma cells compared to a normally healthy, let's say, B cell, right? That wouldn't have a lot of CD20 on it. So your immune system can learn to respond and recognize that there are diseased cells that have this antigen on them. Right? And so you might have anti-CD20 antibody, right, that can be used, right, to defend against this. Right? So in this scenario here, we are not necessarily talking about your body on its own came up with these anti-CD20 antibodies, because in fact, if your body was able to do that, then maybe you would not have developed this cancerous B cell in the first place, right? We are saying that in advanced cancers, right, the very fact that you're talking about cancer means that your immune system has failed somewhere along the way. So if you give high quality antibodies through passively acquired immunity, an infusion of high quality anti-CD20 antibodies, then they can label the target cell and get things like your own natural killer cells to finally pay attention to these cancer cells, right? So the NK cells have Fc epsilon receptor on their surface, and so they can bind to, in this case, oh no, sorry, Fc gamma on their surface, not epsilon, sorry, Fc gamma. And so they can bind to the stems of these high quality IgGs that are being given as a therapeutic, and therefore your own immune system begins to wake up and kill your cancer cells. All right, let's finish up with a discussion about another example of passive immunity, right? Because just in that previous slide, the infusion of pre-made high-quality IgGs that target cancer cells, that's an example of passively acquired immunity, right? Because your body didn't have to make these, you're just getting them as a medication. But the natural way passive immunity usually happens is from mom passing antibodies onto babies. Right? So during pregnancy, IgGs are transported across the placental barrier that separate mom's body from baby's body by the use of the placental cells having Fc receptors on their surface that can bind to IgGs that are flowing by as part of mom's blood, right, and actually transport them through the placental layer into baby's body. Remember, I said this before, but I want to emphasize this. Immunoglobulins are proteins. And just like most proteins, they cannot on their own just diffuse through a cell membrane, right? They have to be transported somehow. Look at this cartoon here. So here is mom before she got pregnant. And this is talking about the distribution of different types of immunoglobulins throughout her body, right? So she has a lot of IgGs, the pink ones, throughout her body. Other types of IgGs like IgE or dimeric IgAs are more focused in regions where there's mucosal layer, right? Like respiratory tract, digestive tract, etc. When she gets pregnant and baby is developing in the womb, IgG is the primary and pretty much only immunoglobulin that gets transported across the placental barrier into baby. What does that mean? That means during gestation, right, mom is actually passing on to baby a library of different immunoglobulins that mom's body has learned how to make. After baby is born, dimeric IgA can be passed from mom to baby through the breast milk. So mom passes on IgG in utero and IgA during breastfeeding, right? So what does that allow you to do? This is an amazing thing, right? I know we're out of time, but I want you to think about this, right? For those of you, the women in this class, someday you might have children of your own, right? And just like any new mom, you're probably going to be really worried about, are you doing the right things to protect your baby against, different types of illnesses, things like that, right? The one thing I want you to keep in mind here, right, is you will be passing on to your unborn child, right, the full library of immunoglobulins that are part of your immune memory, right? So when your baby is born, she will already have these IgGs going through her system, right? And that will be enhanced through breastfeeding, where you are able to pass on high quality IgAs, right? So even though you stress out about doing all the right things as a new mom and worrying, you actually have, right, the power of hundreds of millions of years of evolution backing you up here, right? You will pass on to your child, right, the complete memory of all the different types of illnesses and diseases that you had to fight and survive, right? And come up with IgGs. And you'll provide that to your baby so that she doesn't have to do that at birth, right? Isn't that amazing? Isn't that an incredible evolutionary adaptation? But the problem is, this story is kind of bittersweet, right? Because you are only passing on the immunoglobulins themselves. Where is the knowledge to make more of those immunoglobulins, right? That knowledge stays with the B cells that are in your body, right? So that means even though baby has high levels of IgG in the first few months after they're born, these proteins eventually wear out. So you can see during pregnancy, right? You have passively transferred maternal IgG to baby, but at birth, that's the end of transferring IgGs through the womb, right? So the levels then of those maternally transferred IgGs, those passively acquired antibodies, as far as baby's concerned, begin to fade, right? And after a few months, they're all gone. But importantly, during that time, baby's own immune system is starting up. So you can see this is baby's newly synthesized IgG, right? But this period of birth to 12 months is particularly vulnerable, right? But at least we evolved to provide our babies protection, right? Because if we have been exposed to these pathogens before in our environment ourselves, there's a very good chance that our newly born baby will be exposed to them as well, right? So that we can at least protect them, right, by giving them these immunoglobulins directly. But what I want you to think about is this. This actually reflects where we are in evolution of our species, of our adaptive immune system, right? I said that antibodies represent the newest invention, and our ability then to pass on these antibodies to our newly born children, right, is part of that innovation, right, of how to protect them. But wouldn't it be amazing if sometime well into the future, 100,000 years from now, right, just as long as we don't blow ourselves up first, if somehow our immune system evolves to be able to pass on not just the antibodies, but the knowledge of how to produce those antibodies, then we could be born, right, with true immunity against all of these diseases that surround us, right? Because as I was telling you that story about how your mom passed on IGs to protect you when you were a few months old, I'm sure some of you said, well, wait a minute, that doesn't make sense. Because when I was a little kid, I got sick from all kinds of things again. That's because, right, mom's protection faded after a few months. And then it was up to you to start your own story and build up your own library of antibodies, right, once your immune system started working properly. So that's something to think about. Okay, let's go ahead and stop there for today. Okay.